Great. We have our next speaker, a trainee in the program, Caroline Fleming, who's going to talk to us about examining metabolic consequences of toxicological exposures in resilient urban corals under thermal stress. Take it away, Caroline.
Thanks, Pam. All right, next slide. So first and foremost, I want to introduce everybody to a strange Apoculata, which is an urban coral. Urban coral, maybe not something that everyone has heard of, but a strange Apoculata is a coral that exists up and down the eastern coast of the United States. And it exists in urban habitats such as the New York Harbor. And to prove that to you, there's a picture on the right from Far Rockaway, New York, right near the New York Harbor, of a strange Apoculata. So next slide. So because it is an urban coral, it is existing with urban problems. And so I'm interested really in the effects of this coral and the effects of urban toxicants on this coral. And so two urban toxicants that I'm focusing in on are microplastics and caffeine. So microplastics are really small pieces of plastic, less than 5 millimeters in length, that get sloughed off from larger pieces of plastic and end up in our wastewater, from the clothes that we wear to the things that we interact with. And wastewater treatment plants really are not equipped to filter these out. And so they end up in the wastewater effluent that gets dumped into the harbors. And so microplastics are of particular concern for Astrangia because research from my advisor, Randy Rochin, in the biology department has shown that Astrangia will selectively take up bits of microplastic over its own food. And so it's definitely a potential issue for these corals. So I'm interested in the effects of microplastics on this coral, but also the effect of caffeine. Caffeine is a ubiquitous urban pollutant, something that I drink, and I know many of people in the audience drink every day. And so, but it's also a metabolic disruptor. And I'm really interested in the effects of these toxicants on the metabolism of this coral. And so we're going to be looking a little bit more into that. Next slide. So I would be remiss if I did not include human health here. So Astrangia exists in the same areas as really important shellfish species, such as scallops, mussels, oysters, and the like. And it exists in urban ports such as New Bedford Harbor. It doesn't quite make it to Boston, but Astrangia exists in New Bedford Harbor, which is, I think, self-proclaimed America's #1 fishing port. And so I don't know about you, but I would really rather not have bits of microplastic in my oyster that I'm having from New Bedford Harbor. And so we wanted to think about different interactions of those toxicants with these marine invertebrates. Just to make things more fun, we wanted to layer the effects of increasing sea surface temperature because the oceans are rising, or the ocean temperatures are rising. And so we want to look at those interactive effects of how these toxicants are going to affect the metabolism of corals under these higher sea surface temperature regimes. Next slide. And just to make things even more interesting, we're interested in the effects of these toxicants at different life history stages. So hopefully by now I've introduced you all to the adult coral, but next we have the egg, and next we have the larvae. And so I'm thinking about all of these different potential life history stages and how urban toxicants might affect these different life history stages differently. Next slide. And so in summary, our guiding question is, how do increased temperatures and these pollutants, specifically microplastics and caffeine, affect metabolic stress in a strange Apoculata from New York Harbor at these 3 different varying life history stages? And our hypothesis is that increased temperatures are going to interact with those pollutants and cause increased metabolic abnormalities, with those early life history stages, just because they're so small and fragile, being more vulnerable. And I'd like to do lots of acknowledgements, but thank you all for your time.